When UT became an official university in 1883, a biology program, much less a herpetology collection, was not on the agenda. It would be about half a century before the herpetology collection had its first catalog specimens, and another 50 years or so would have to pass before UT would become a herpetology juggernaut. The first herpetology work at UT occurred in the early 1900s and was done on campus. The focal species of that research is now recognized as Sceloporus olivaceus, the Texas spiny lizard. A paper on this lizard was published in 1909 in the Bulletin of the University of Texas and was written by Horatio Hackett Newman and John Thomas Patterson. At the time, Newman was the new chair of the School of Zoology and an embryologist interested in the processes of multiple births. Patterson was a new instructor at UT, an embryologist himself, but would later become a driving force with genetics research on campus, as well as department chair. This paper was an observational field study of the Texas spiny lizard, expanding what was already known about the lizard living in captivity. At the time, the site of the study, the UT campus and surrounds, were very different, meaning there were way fewer buildings and much more mesquite. On campus, Newman and Patterson observed the lizard's daily behaviors and paid special attention to the role trees played as sources of food, protection, and sunbasking. From this point on, not much happened in the world of herpetology at UT. That would change with the arrival of William Frank Blair in 1946. He was the first to have a lasting impact on herpetology research at the university, as well as to significantly add to the specimen collection. Blair was born in 1912 in Texas, but his family moved to Oklahoma in 1916. As an undergraduate, he studied zoology and botany at the University of Tulsa, earned his master's in science in zoology from the University of Florida, and his PhD in zoology from the University of Michigan. After serving in World War II, Blair accepted a position at UT in 1946 and was promoted to professor in 1955. Blair's initial research focus was on mammals, particularly rodents, with interests in ecology and distribution centering on the concept of a home range. With several colleagues, he wrote the widely used Manual of Vertebrates. Blair's interests, however, would soon shift to amphibians and lizards. In Austin, Blair and his wife, Fern, bought a 10-acre property where he and many of his graduate students conducted quite a bit of research. Here he carried out studies of amphibian reproduction. During one of the worst droughts in Texas history, from 1952 to 1956, he and his students observed over 3,000 individual Texas spiny lizards for a period of 5 years. This resulted in the highly influential publication, The Rusty Lizard: A Population Study, from 1960. It was one of the first studies that calculated age-specific fecundity, which means maximum reproductive potential over a lifetime, as well as survival for a species. In the 1950s, Blair initiated a program to study whether different toad species were able to produce reproductively viable hybrids, meaning their offspring were not sterile. They had some success providing some measure of relatedness between the two parental species. Blair's focus soon shifted to Bufo, a genus of true toads in the amphibian family Bufonidae. With his colleagues, he would analyze factors such as hybridization potential, mating call, breeding periods, serum proteins, parotid gland content, karyotype, skull morphology, and laryngeal structure. He also studied the parallels of Bufo in the faunas of desert regions in North and South America. This led to his involvement in the International Biological Program, a 57-nation project sponsored by the International Council of Scientific Unions. The hybridization data Blair generated is still in use by researchers today. And what about the early years of the herpetology collection? Fortunately, that was a focus almost as soon as Blair was hired at UT. He founded the Texas Natural History Collections, now the Biodiversity Collections, in the late 1940s. The earliest cataloged herpetology specimens date to 1939. Specimens received in exchange from the University of Florida, with just a few others collected in the early to mid-1940s. Initial collection growth outside of this coincided with the large-scale trips organized by Blair between 1948 to 1955 to West Texas and the Panhandle, as well as many local and regional collecting trips by Blair and his students. In the 1950s, Blair noticed that the mating call of frogs and toads played a role in mate selection. This research will be the focus of our next video in the history of UT herpetology.